So we'll move on to our second keynote presentation by Dr. Philip de Jager, who's a clinician scientist and chief of the immunology division at Columbia University Irvine Medical Center. He studies the onset and course of neuroinflammatory and neurodegenerative diseases, and his lab uses cutting-edge functional genomics, spatial multiomics, and computational approaches to dissect cortical cellular microenvironments and identify molecular drivers of trajectories underlying brain aging, neurodegenerative diseases, and cognitive resilience. Phil, welcome, and we look forward to your talk.
Thank you, and it's a real pleasure to be here with you today. So without further ado, let me proceed. So these are my disclosures. They're not pertinent to this talk, except for the last one. I'll be showing one example of a collaboration with industry with a vaccines company, but we have received no research support or other support from this company, and I have no interest in the company, just sharing data. So the challenge that we set out to address is the following. We want to answer the question of whether we could use a data-driven approach to characterize neocortical cell states and uncover new insights into brain health, aging, and that would then lead to therapeutic implications. And my origins actually are in human genetics, statistical genetics. These are a powerful tool for gene discovery. But it's clear that, of course, there's a lot that happens during development of the brain and also during the life course. And these dynamics are not captured by human genetics. And so how can we capture the cellular interaction of an organ that in humans we, of course, can't access during life? So these were some of the challenges that we were facing in our project. The case that I'll sort of share with you today is a project that we've pursued. There have been a couple others of this scale, and they, I think, have had led to similar results. But I think we have one interesting sort of closing the circle back to therapeutic development that I'll share with you today. And so we're focusing here on the trajectory of aging in the human brain, which often leads to the accumulation of amyloid proteinopathy. So the emergence of amyloid aggregates in different parts of the brain. This in turn typically leads to a second form of proteinopathy caused by the tau protein. These are called neurofibrillary tangles. And again, there's a lot of nuances. These are just sort of the most common sort of trajectories. And ultimately, temporally closely related to the accumulation of tau proteinopathy is the symptomatic manifestation of these events, which is cognitive decline. And I've been part of multiple different consortia that have been attacking this question using high-dimensional data. First, the Accelerated Medicines Partnership for Alzheimer's Disease, AMP-AD, the Functional Genomics Consortium for Alzheimer's Disease, and most recently Cascade, which is a new program that's focusing on spatial analysis of the aging brain and Alzheimer's disease. But today I'll be focusing on some of the work from the AMP-AD consortium. And so again, we have these different stages of the disease that are recognized and sort of are the paradigm of the disease. But what are the causal chain of molecular and cellular changes that are driving these transitions? So we took an approach, again, that's now quite common. So here, the data I'll talk about today is a protocol that we started with Viv Regev close to a decade ago now. And we applied this to a collection of brains from the Ross and MAP cohorts based in Chicago. These are very well characterized individuals, antemortem, and then they prospectively donate their brain, and then there's a detailed pathological examination of the brain afterwards. We focused on the dorsolateral prefrontal cortex, and as you heard earlier, really in humans, it's very important to have a large sample size. And so today I'll be sharing you with your results from 869 participants in the ROS-MAP collections, where we've generated 4.2 million transcriptomes from single nuclei isolated from these brains, from this frontal cortex. And so you can see here at the bottom, the typical sort of UMI and UMAPs, and you can see the different cell types that we've uncovered, again, very sort of standard. So, and as you've heard, you can subdivide these cell types into cell states or subtypes for each of them. And so from the 7 major cell types, we identified 96 different cell states or subtypes, depending on the cell type. And then we asked a very simple question, which is, which of these cell states are are influenced by or associated with the disease. And so we looked at 3 different traits. Each trait here is shown on the left side in the panel. You can see that there are 3 traits: amyloid first, then tangle, and then the slope of cognitive decline. And in rows, we have the different cell states. And so you can see that the most extreme results are for microglial 13, which is a subtype of microglia. And for the aficionados, this is enriched for the signature from the disease-associated microglia from the defined originally in mice. We also find a second microglial subtype that's also strongly associated with all 3 traits, one oligodendrocyte subtype and a subtype of astrocyte called astrocyte 10. So these are they stand out in terms of their association. As you can see here from other states, and these are not all the states, these are just sort of a selection of the ones with the highest associations. And interestingly, the direction of the effect is that microglial 13 and astrocyte 10 both increase in frequency with worsening disease. And the other thing that I would like to note is that both of these pathologic cell states are relatively rare. So if you look at microglial 13, they represent about 1% of all microglia. In the context of disease, this frequency goes up to about 1.5%. So it's a small shift, but it's highly significant, of course, because we have a large sample size. Similarly, in astrocytes, the astrocyte 10 population is actually relatively small, less than 0.5% of astrocytes in healthy individuals, and but the frequency goes up about doubles in some of the people with Alzheimer's disease. We also sort of had noticed that if you look at patterns of these cell types across individuals, so we have these extreme results of different cell states associated with disease, but they're not working by themselves. So we saw that the frequencies of these different cell states are correlated across individuals. And so we were able to define different cellular communities that bring together different cell states. And presumably these different associated cells that are part of a community are talking with one another and sharing some signals that are then driving the disease. One of them is shown here, which again, these are the top results of the association study that I reported in the prior slide. And they form a separate community that we call C2.3, and this is the one that really captures all the association with Alzheimer's disease. But this still doesn't tell us exactly which cell state may be having a role at which stage of the disease. So for this, we used a structural equation modeling to try to infer from cross-sectional data, and this is important. So this is not a causal model since we don't have longitudinal data. We are limited to what we can capture at autopsy. But here we build a model about where these different cell types are maybe having an effect in the causal chain of events leading to cognitive decline. And what we propose, again, and this is our colleague, Hyun-Sik Yang at the Brigham and Women's Hospital, who did this part of the work. And so he did this nice model where we show that microglial subtype 12 seems to be involved in the accumulation of amyloid proteinopathy. Then microglial 13 seems to be involved in the transition from amyloid to tau proteinopathy. And finally, and to me as a clinician, most interestingly, astrocyte subtype 10 may have an important role in the transition from the accumulation of tau proteinopathy to the emergence of cognitive decline, the sort of symptomatic manifestation of disease. Oligodendrocyte 7 may be a downstream effect, so maybe a reaction to some of these changes, so that this remains to be determined further. But basically what we've done is to prioritize the different cell states that may be related to different parts of the trajectory of the disease. The other thing we have done is to take the cell frequencies for the R96 cell states and then project individuals here. So here the dots are individuals, and to create a manifold of where these individuals lie in this multidimensional space. And very interestingly, so the solution here was best for 2 trajectories, one of which is shown in orange and the other in blue. Importantly, I have to emphasize that the starting position at the bottom right is actually there's a fair number of individuals who are there. And so this, I would say, captures individuals who die and who are cognitively non-impaired and have minimal pathology. And so these are what we can expect are relatively homeostatic older brains. Of course, nothing they have changed, but they haven't had much pathological effects. And then we can see that one trajectory has individuals growing towards an endpoint that goes that goes north, and that's we call this a progression to Alzheimer's disease, because as you get further along this trajectory, you will have more amyloid first, more tau, and eventually more cognitive decline and dementia as you go further down this trajectory. And interestingly, astrocyte 10 is the cell state that really defines the endpoint of that trajectory. Very interestingly, we have an alternative trajectory. So other individuals go towards the left, and they are going to this other alternative endpoint that's defined by a different astrocyte state, in this case, astrocyte state number 5. We've looked actually quite hard to try to understand where exactly this trajectory may be related to, but we haven't found any association so far. So there is some dementia, but it's not necessarily at the tip of the trajectory, unlike the progression to Alzheimer's disease. And we haven't found any association with Parkinson's traits, with vascular disease or other traits that are captured neuropathologically. So that remains to be determined. So I want to explore a little bit more this astrocyte subtype number 10. And so here what we did is to try to explore a little bit further the relationship between astrocyte 10 and the slope of cognitive decline in these older individuals. And so what we did here is to have one model that has all the different variables that are known that have been previously reported to affect cognitive decline. And those are shown in rows over here, and you can see the effect size of each of these traits in the forest plot. And on the left side, we have the basic model, and then on the right side, we add the astrocyte 10 frequency at the bottom. And you can see here that we have highlighted in blue those traits that remain significant in explaining some of the variants of cognitive decline after accounting for all the other variables at the same time. And so quite interesting to us, astrocyte 10 frequency, even when you account for all the amyloid, the tau pathology that define Alzheimer's disease, Lewy bodies that are related to Parkinson's disease, TP43, vascular disease, even when you remove all these other known pathologies, astrocyte 10 frequency still explains some of the residual cognitive decline that these individuals are displaying. And so this is quite interesting to me and suggests that astrocyte subtype 10 may not be specific for Alzheimer's disease and may serve as a common endpoint, for multiple different neuropathological processes that ultimately affect cognitive decline. Since we had proteomic data from the same brain region of the same brains, we're also able to ask a different question, which is, how does astrocyte 10 work exactly? How is it affecting cognitive function? And we used SNAP25, but we've also done this with other synaptic proteins as a proxy for synaptic bulk in each of the brains. And what we show in the mediation model is that actually the expression level at the protein level of SNAP25 explains some of the effect of astrocyte 10 on cognitive decline. So again, this suggests that one way that the astrocyte 10 state may be affecting cognitive decline is through synaptic loss. So we now have a mechanism and a state that's implicated in the disease. So we wanted to take the next step and to try to understand, okay, why do astrocytes adopt this astrocytic state? Because this would be this first step to try to understand what may be therapeutic targets that we could pursue. So we used a method called NicheNet to try to infer what ligands and receptors may be driving the astrocyte 10 signature, in this case, using astrocytes as a receiver cell and other astrocytes or microglia as the sender cell. And so we did this, and we prioritized, and here in rows to the left on panel B, you have the different ligands that are prioritized, and then you can see the little dots, and each of those columns are a different astrocytic state. You can see at the bottom, the astrocytic state are highlighted in green, and in purple, you have the microglial states. And you can see that different cell states different ligands are associated by different cell states. To the right, we have the same analysis for the receptors. And again, each row is a different ligand in this case. And then on the right side, on the columns, we have the receptors for that particular ligand. Okay, so we now have ligand receptor pairs that we've implicated, and we can do some further work to try to prioritize which are the ligand receptor pairs that are best explaining the presence of the astrocyte 10 state amongst our single nucleus data. And so we've highlighted in purple here, in pink rather, at the top, 5 of the ligand receptor pairs that are they explain the most variance in the frequency of astrocyte subtype number 10. And we've actually gone on to, and this is on bioRxiv for those who are interested, we've gone on to validate 2 of them, semaphorin 4D and the plexamine 1, as well as JAM2 as another ligand. And here I'm going to focus on semaphorin 4D, which is shown here in the crystal structure, where we have a semaphorin 4D in green, which is a soluble protein, and it binds to plexin B1, which is on the surface of astrocytes. And then, of course, downstream for this, it triggers the astrocyte 10 signature. And we've been able to reproduce this in vitro, both for semaphorin 4D and for JAM2. And then we took a different approach to try to say, well, how can we validate this further? So we did a relatively simple analysis, repurposing some Visium data. And so Visium, of course, is a spatial transcriptomic platform. And this is the old version, which has spots about 50 microns, which contains about an average of 4 cells per spot. And so we identified those spots which had a high astrocyte 10 signature in the tissue section. And then we basically asked the question as to whether the neighboring spots around, which are shown here in purple, around the one in the center that's yellow and that has the astrocyte 10 signature, those spots should logically have a higher expression level of the ligands if our model is correct. And this is what we show here on the left side. There's a strong correlation between the two. And if we take random spots from all throughout the section, we don't see this correlation. We've gone on now to use a different platform, Xenium, to do this even at the single cell level, and those results are consistent, but I don't have time to present them today in the interest of time. I do want to sort of have one more result, which is quite interesting. By chance, actually, we met a company that had produced an anti-semaphorin 4D antibody that they were testing in older individuals. And so we repurposed their phase 2 clinical trial data where they had spinal fluid from individuals collected before and after treatment. The treatment was for 48 weeks. They had done actually a proteomic profile of these spinal fluids. And so we simply derived an astrocyte 10 signature derived from our RNA data in the CSF proteomic data. And some of the results are shown here. To the left, you can see that the prevalence of semaphorin 4D level in the spinal fluid at the protein level correlates very nicely with our astrocyte 10 signature for the proteins that are found in the CSF that belong to that signature. It also relates to GFAP, which is a general marker of glial activation. And most exciting, to the right, we see the results. So in terms of the therapeutic intervention of this blocking antibody, and if you block semaphorin 4D, you can see that the people receiving the treatment are in blue. In red, we have the placebo-treated individuals, and you can see that the astrocyte 10 signature is reduced in the individuals that have received the treatment after 48 weeks of treatment. So with that, I'll sort of close the presentation and just highlight a few things. So I think we've shown that we can have a data-driven approach to the human brain that yields robust, reproducible results. I think that's very important, again, highlighting some of what we heard in the previous presentation also, that these cell states are reproducible across different data sets. We have now analyzed a large number of brains, and we've identified 2 different trajectories shared, still trying to understand exactly what this alternative brain aging trajectory may be. But we've highlighted astrocyte 10 state as a key state for Alzheimer's disease, and most likely for other neurodegenerative diseases as well. So that's another further question to pursue. And again, I showed you that we can go from there to identify putative drivers of these astrocytic states of interest. And in this case, semaphorin 4D, and if we intervene, we seem to have target engagement with the astrocyte 10 signature. And I think the one remaining challenge for these types of studies is that we don't really yet have a community-based taxonomy, and we need to get this to really speak the same language as we do these analyses. With that, I'll just highlight the team. This is a very large team, of course, with many collaborators. Particularly Natasha Comandante Liu, who's a postdoc who did the beautiful work on semaphorin 4D and the niche net modeling. Thank you.
Great. Thanks very much, Phil. We have time for one question if you want to raise your hand. Dr. Zhang?
Right. This is a very exciting kind of development in understanding the mechanism and trajectories, and so there are two general ones. Can you put some suggestion or thinking of how one may find from the population level in real world data from EHR, CMS, in those data sets, how one may be able to correlate or observe your findings? What would be the strategy you suggest?
This is a great question, and we've been exploring it. The challenge is that you need, of course, the both types of data, and this is where the collection that we work with, Ross and MAP, have a lot of antemortem data, and so we're trying to find correlates of these trajectories in the antemortem data to see if we could predict while somebody is alive whether they are on one or the other trajectory. So far, we haven't had much success, and so I think this is where we require more work and a finer definition of these cell states. I think this probably gets to some of the questions that we would like to explore in this meeting, which is that we have defined arbitrarily these different cell states for our study. But I think if we can begin to refine them across also brain regions, as we heard in the previous talk as well, this may help us to have a CRISPR signature that we can then pursue in blood and in spinal fluid, perhaps.
Great. Thanks very much to all of our keynote speakers. In the interest of time, we'll stop there. Please feel free to use the chat function to continue the conversation.